Greetings to all the students. Welcome to biology and chemistry lessons. We are once again here with a new biology topic, osmosis. So what is osmosis? Osmosis is the diffusion. Last class we have seen in our biology class how diffusion takes place in living cells. In case of the human beings, how the different molecules get in and out of the cells uh, across the semi-permeable cell membrane from high to low concentration. Osmosis is also a diffusion, but it is slightly different from diffusion. How? Osmosis takes place only when the water molecules move. All right, it is only the diffusion of water molecules from high water potential to the area of low water potential. High water potential means where the concentration of water is more, where water is more. Water will move from the area where it is more towards the area where it is less, provided there is a semi-permeable membrane, living or non-living, in between the two concentration areas. So, how it is slightly different from diffusion? In diffusion, every sort of the liquid and gas molecules move from the area of high to low concentration. There can be a semi-permeable membrane, there may not be a semi-permeable membrane. Diffusion takes place. But in case of osmosis, only the water molecules will move. Only the water molecules will diffuse from the area where water molecules are more towards the area where the water molecules are less, but there must be a semi-permeable membrane. Just like diffusion, it is a passive transport. It means no energy is needed in this process. Why it is a passive transport? Because whenever the particles are moving, whenever the molecules are moving towards the concentration gradient, what does it mean? From the area of high concentration to low concentration, whenever the movement of the particles takes place, we call it a passive transport, and it does not require energy. Now, you people will think some of the differences in between diffusion and osmosis. So this is about diffusion, that water molecules move high to low concentration. Always semi-permeable membrane must be there. It is a passive transport. It takes place towards the concentration gradient. Now we will see what are the conditions necessary for osmosis. The conditions which are necessary for osmosis to take place is concentration gradient and the semi-permeable membrane. What is the meaning of concentration gradient and semi-permeable membrane? You can see in this container, right in the middle, there is a semi-permeable membrane. On this side, the water molecules, these blue color tiny molecules representing the water molecules, you can count them, you can see them, they seem to be more. But in this side, you will see that the water molecules are less, but the sucrose molecules are also there. We have dissolved some sucrose or some salt in this side of the container. So the water molecules are less. So what will be the direction of the movement of the particles, and which particles will move? Only the water molecules will move from the area where they are more towards the area where they are less. If you will remove the semi-permeable membrane, then it will not remain osmosis anymore. Then what it will become? It will become simply diffusion. Then who will move? Then the salt will move from the area where it is more towards the area where it is less. So this is the difference in between osmosis and diffusion. Now, we will see what are the different type of the solutions. So for example, first of all, what is a solution? A solution is a mixture of solute and solvent. What is a solute? When you dissolve some salt or sugar in a glass of water, salt or sugar is the solute and water is the solvent. There are three different type of the solutions. One solution is called a hypotonic solution. Hypo means something which is less. So in this type of the solution, the solute, the sugar or salt is less and the water is more. Or you can also call it a dilute solution. Hypertonic solution. What is the meaning of hyper? Something which is more. So in this type of the solution, the solutes, the sugar or salt is more in water. Or in other words, you can say this is a concentrated solution. This solution is a concentrated solution. So remember hypo and hyper. hypotonic solution. Hypotonic solution is a very weak solution, dilute solution, more water, less solute. Hypertonic solution is a very strong, concentrated solution with more salt or sugar and less water. You can see the example over here. Here, right in the middle, you can see that this is the semi-permeable membrane. All right, so this is the semi-permeable membrane. So you can see in this area and in this area. Where water is more and where water is less, you can see this blue color area is, water, and here is the salt solution. So this area is the hypotonic solution. These two areas, you can see the number of the molecules are more on either side. So they are isotonic solutions. In case of hypertonic solution, you can see in this area, the molecules are more, and in this area, the molecules are less. So this is the hypertonic solution. So hypo, hyper, and isotonic solutions. Now we will see how this osmosis works in animal and plant cell. So this is one of the plant cell. When you will dip the plant cell in water, when you place the plant cell in water, we all know that plant cells are surrounded by a cellulose cell wall. So this is the cell wall. So you have this surrounding is the water. So you have placed the plant cell, this plant cell in water. Inside the plant cell, there is a big vacuole, and the vacuole contains a cell sap. What is a cell sap? A solution of mineral salts which is coming from the soil. Outside, the water is more. Inside the cell sap, the water is less. So osmosis will start happening and water molecules will move from the area where they are more to the area where they are less. You can see in the second condition how the vacuole has become big. The vacuole has become too big. So such type of the cell in which there is a big vacuole is called a turgid cell. The cell has become stretched. Inside the fluid, large amount of fluid is accumulated, and the turgor pressure, the pressure which the fluid is exerting on the cell walls, the turgor pressure inside the cell increases. Such type of the cell is called a turgid cell. So what is a turgid cell? A turgid cell is a cell in which water has been gained from the surrounding by the osmosis. The vacuole has become too big. The fluids are exerting pressure, the turgor pressure on the cell wall, and the cell has become stretched, increased in the volume, increased in the size. This is called turgidity, turgid cell, and it is due to the osmosis. But the cell is not going to burst. Why the cell is not going to burst? No matter how much water it accumulates from the surrounding. But the cell is not going to burst because there is a protective cell wall all around it. So now see, when a plant cell is surrounded by water or a hypotonic solution in which the water molecules are more outside and less inside, water molecules move inside the cell by osmosis across the semi-permeable cell membrane. The cell accumulates water and becomes turgid. The turgor pressure inside the cell, that is the pressure due to the accumulation of the fluid, increases and the cell bulges out. The cell does not burst because of the cell wall. Now see the example of the plant. You can see in the soil, the potted plant, this is the soil, and you have watered the soil. You are giving regularly water. So water is more in the soil. Now you take the example of this root here. This is a single root here. Inside the root here, there is the cytoplasm. So cytoplasm is a concentrated solution of mineral salts. Outside of the cell, root hair cell, there is a semi-permeable cell membrane. So water is more outside the root hairs and water is less inside the cytoplasm of the root hairs. Osmosis will take place and water will start getting inside the roots. This is how the plants absorb water. And then this water moves from one cell to other, from one cell to other by osmosis. And it will be spread throughout the plant. As a result, each and every cell of the plant body becomes turgid. The turgor pressure of every cell increases. And as a result, what will happen? The plant will get the internal support and plant will be able to stand erect because every cell inside the plant cell has become inflated, has accumulated lots of water. So the plant can stand erect. So whatever the plants and the trees you are seeing around you, those who are standing erect by themselves without any support, it means they have got all the cells turgid. Osmosis is taking place and the water is continuously getting inside from the soil into the roots. So this is the benefit of osmosis. Now, the effect of osmosis in the animal cells, human cells, we do not have any cell wall. So if our cells are placed in water, definitely osmosis will take place. Water molecules will move across the cell membrane inside our cytoplasm and our cell will become turgid and eventually the cell will burst because there is no cell wall. Now, opposite of osmosis, what happens? Opposite of osmosis is called plasmolysis. So what is plasmolysis? You are placing a plant cell or animal cell in a very concentrated solution of salt or sugar. It means outside the cytoplasm, the solution is very much concentrated and comparatively the cytoplasm of the cell has become dilute. So water will always move in osmosis from the area of high to low concentration. So water will move from the area where it is more towards the area where it is less. So water will move from inside to outside. Water will leave the cells and as a result the cell will shrink away. So negative osmosis is taking place. Osmosis is taking place in the negative direction. And the negative osmosis is called plasmolysis. What will be the effect of plasmolysis? If a plant cell is surrounded, you can see this effect. This is the red color cells. You can see this is actually the microscopic picture of the onion cells, onion epidermal cells. That is a thin membrane in between the onion scales. So we will take out this layer and we will put it in the dye so that the cytoplasm turns red and we can see it properly under the microscope. So you can see in the first picture, this is the picture of an onion epidermal cell. So over here, osmosis has taken place. You have placed the onion epidermal cells in water. So water has got inside, entered inside. And you can see the cytoplasm is nicely spread. All the cells are turgid. This one is plasmolysis. It means you have placed the onion epidermal cells in the concentrated salt or sugar solution. So see what will happen. Inside, the concentration of water was more. Outside, it was less. So water molecules are coming out of the cell and you can see as a result what will happen. The cytoplasm shrinks away. Cytoplasm will leave the cell membrane. Gaps are created in between the cell membrane and the cytoplasm. The vacuole has become very small. The cell will shrink away. Such type of the cell is called a plasmolyzed cell. A plasmolyzed cell, the turgor pressure falls. Inside, the fluids are less, so turgor pressure is less inside the cell. As a result, you can see here all the cytoplasm in every cell, they have left the cell membrane and gaps are created. This white portion is actually the gap. So this is bad for the plant. Why it is bad for the plants? Because when all the cells of the plant for some reason have become plasmolyzed in the soil, when water is less and salts are more, from the root hairs, water will come out by plasmolysis. And when all the cells of the plant will shrink away, the plant will not get the internal support. So what will happen? Drooping. The plant will not be able to stand erect. Eventually, the plant will fall down. And this falling down of the plant is called wilting. So what is wilting? Wilting is a phenomena in which plasmolysis has taken place and water molecules are drawn outside the cells. And all the cells in the plant, they are shrinking. Plant is not getting enough support to stand erect. So the plant will fall down. So this is called wilting. The falling down of the plant because of the plasmolysis is called wilting. So this is a real life picture. You must have noticed one thing. If you will not water your plants for a long period of time, or if it is not raining for a long period of time, you will see that all the leaves of a plant will become droopy. The leaves will not be able to spread themselves the way they have to be. Why? Because they are losing water. Plasmolysis is taking place. the cytoplasm is shrinking away. The cells are losing their turgidity, and so all the leaves will become droopy. What will happen if plasmolysis takes place in the animal cells in our cells? Our cells will also shrink away. And if this condition will not be restored, our cells will permanently damage. If our cells are placed in salt solution, water molecules will come out of the cells from high to low concentration and the cells will damage. But usually it is not happening in case of the animal cells. It is particularly plasmolysis is the phenomena which is taking place in the plant cells. So what is the example of the osmosis in animals, example human beings? From our intestine after the digestion, the liquid food when it reaches to the intestine, water goes from intestine into the blood by osmosis. In our kidneys, whatever the water is extracted or filtered by the kidneys will return the water back to the blood. Most of the water will be returned by the kidney back to the blood by osmosis. What is diarrhea? Diarrhea is a particular example of, you can say, plasmolysis. So whenever we have a bacterial infection in our stomach, so in our stomach, bacteria will start producing the poisonous toxic substances due to which the salts come out of the cells into the intestine. So when the salts start coming out of the cells into the intestine, whatever the liquid food is there in our intestine, the concentration of that liquid food increases. It becomes a hypertonic solution. And if you will compare it with the blood vessels are surrounding the intestine. So the blood inside the blood vessels becomes very much dilute. And the liquid food inside the intestine or the stomach becomes very much concentrated. Negative osmosis or plasmolysis takes place. Water start coming from the blood into the intestine. As a result, we suffer dehydration and that results in the watery loose motions. So there are some experiments of osmosis. So you can see we have taken a thistle funnel over here, and in the thistle funnel, the end of the thistle funnel is wrapped with the artificial semi-permeable membrane, and inside the thistle funnel we have taken sugar solution and we have placed the thistle funnel in water. So now you can see water is more in the beaker outside and water is less in the thistle funnel, and there is a semi-permeable membrane over here. So water will start getting inside by osmosis. And as a result, in some time, we will see that the level of the glucose solution will rise. And if you will not remove it, you will see that the glucose solution after some time will start overflowing. Why? Because osmosis is taking place and osmosis takes place very fast, very quickly. It is not like that we have to wait for some time. No. This is another example. You cut some sticks of potato and here in this glass, let's say in this glass, you have taken a very concentrated salt solution. And in this one, you have taken only water. So you have placed the potato sticks here and here as well. And then after some time, you will remove, and this is the result in front of you. All right, before doing the experiment, you have to measure the length of each potato stick. And then you will remove them after some time. You will dry them with a tissue paper, measure the length again. In this one, the length will decrease and it will become little bit loose, floppy. In this one, the length will increase and they will become very much stiff. Now you have to think the reason why it is happening. Think about the solution outside the potato and the cytoplasm inside the potato. Compare these two solutions and guess where osmosis is taking place and where plasmolysis is taking place. So you will be able to get the answer. Now we will see another experiment. So in this experiment, this is actually a cutting of the question paper. This was the question in the question paper. This is a potato. All right. So this is a potato. We have made a cavity in the potato. And we have taken some sugar in the cavity and we have placed the potato in a beaker of water. And after some time we have seen, you can even do this experiment at home and you can see the result in no time. You will see in some time, water has risen up in the cavity and water has dissolved the sugar in it. Why? When you have put sugar inside it, when you have put sugar inside the cavity, compare the concentration inside the cavity and outside. Outside water is more, inside the cavity water is less. So water will start moving from the area where it is more to the area where it is less. So gradually, slowly, one cell after another, one cell after another, the cell will reach to the cavity and the cavity will fill up filled up with the water and sugar will be dissolved in. So this is one of the typical question comes in the question paper. That's all for today, children. I hope you have understood and enjoyed the topic. Bye bye. See you in the next class.